Okay, could somebody confirm you can hear me?
I can hear you.
Thank you. Before I share my screen, let me ask her first, are there any questions?
When are you going to be emailing us the unknowns?
Information? Hopefully tonight.
Okay.
Hopefully it'll be after the lab, but hopefully tonight. I'll need to send out your unknown number and your first test results tonight. That's my plan. I'm not ready, but hopefully I'll be ready tonight. Before we get going and before I share my screen, let me state that I've done a fair amount of grading. And so I'm calling up the grade book, which is why I'm not sharing my screen. I got some of the extra credit steady graded. I think extra credit steady 5 and extra credit steady 3. I got worksheet for lecture 3 graded. I got worksheet for lab 5 graded for the first time. Nothing has been graded the second time. I got practice quiz 00B, the multiple choice questions graded, and actually quiz 00A, the practice quiz 00A, I graded earlier, but you have to click on the, I don't know, the grade or the file to see the grade. For some reason, this quiz is not showing the score until you click on it. And you'll note that this is not being added to your current points, but it is being added to your total points. And I've ran into this problem before. I think it's a bug in Canvas. And for some reason, this practice quiz 00A, the grade will not be showing, but if you click on it, you can see it. And I think that's it. So take a look at your grades. If you have any questions, you can either ask or email me. I need to go into quiz 1C and quiz 2C to see if I'll be adding any extra credit. I normally do that a little earlier now, but I'm a little behind schedule here. So I will look into that. I think, let me see, quiz 1C. I think that one actually had a question that said it was an extra credit question. It's not this first question, so I'll have to look through that. I'm not going to look through it now. All right, any question about the grades? If not, I'm going to get out of the grade book and begin the lesson. Sharing my screen now. You should see my screen. So today is February 12th, and we are supposed to be on chapter 5. We are. Do let me warn you that you have a quiz this Wednesday. The quiz will be quiz 3, and it'll be covering labs 5 and 6, as well as chapter 4 and the end of chapter 3. Any questions about the quiz? Or any questions about what we're doing? There is a lab tonight, and it will be lab 7. All right, we were talking about enzymes and I had shown you the slide where enzymes reduce the activation energy. Is this not where somebody left off? Because this is where I've got a note that we left off here. So enzymes are biological catalysts. They speed up chemical reactions. We can speed them up by, you don't need to know the numbers, but 10 to the 8th times up to 10 to the 10 to the 10 fold times. Enzymes act on specific substrates and they are encoded by genes. So our genes encode and program the enzymes. Enzymes are specific for a single chemical reaction. This is a little bit of an oversimplification. Those of you who have know anything about medicine or you've worked in the clinic and they use enzymes, we do have some drugs which mimic a substrate and they will compete with the substrate for the enzyme. So it's a little bit of an oversimplification when we say that one enzyme works specifically on one substrate and that you have one enzyme for each different substrate. A little oversimplified, but we're going to keep it nice and simple in this class. And we'll just briefly talk a little bit about the drugs and I'm just going to make it a side note, like when we're talking about, what is that called? Um, inhibitors of enzymes. We will mention just a little bit about drugs, but other than that, we're not going to talk about that topic. Enzymes have their specificity to one specific substrate because of the enzyme's three-dimensional shape. And then there's a portion of the enzyme which actually binds to the substrate, and that we call the active site of the enzyme. So the active site must fit the substrate. And like I said, it is a little oversimplified, but in this lesson, we're going to say that one enzyme specifically acts on one specific substrate. Enzymes are not used up or altered in a biochemical reaction. So when they finish with that reaction, they can bind to another substrate and perform another biochemical reaction. Enzymes, when they bind to a substrate, they will form an enzyme substrate complex where the two are literally bound to each other. This orients the substrate to increase the probability that the substrate will change into the product. So if you think about a large substrate, you can put stress on the bonds of that substrate when the enzyme is bound to the substrate. And then when you put the stress on those chemical bonds, it will break those chemical bonds and convert the substrate into the product. Now, of course, that's just one way of looking at it. You could also look at it, you've got two smaller substrates, and the stress that the enzyme puts on them makes those two substrates form a chemical bond between them. But the idea is that the enzyme substrate complex orients the substrate to increase the probability of the reaction occurring, and this lowers the activation energy, making the substrate change into the product. Enzymes do have a specific name. If a word ends in ASE or ACE, that's the naming convention for naming an enzyme. Here are a few of the different enzymes. We'll talk about ligase, and then there's one on here that isn't mentioned. We'll talk about catalase in the lab. We've probably already talked about that one. We will talk about these enzymes, and they end in ASE. This is the naming convention for naming an enzyme. Now, not all enzymes end in ASE. Most of the enzymes that do not end in ASE are enzymes that were named long time ago before the naming convention was ever proposed. Like renin is an enzyme in our stomach that curdles milk, and it's really important for babies to curdle the milk, and then the milk will stay in the stomach longer, and so that the stomach has more time to digest the milk protein in particular. So renin is an enzyme. It doesn't end in ACE, but renin was named a long time before the ACE or ASE naming convention was proposed. We should mention that enzymes don't work in a vacuum. Some enzymes require a helper. This helper component is either a coenzyme, which is a small biological molecule, or a cofactor. Most of the cofactors are a element like iron, zinc, magnesium, calcium. The cofactor, the coenzyme, actually binds to the enzyme, making the active site of the enzyme. And we're not going to talk about, I'm not going to ask you what's the name of an enzyme before the helper binds to it, and then what's the name of it after the helper binds to it. We'll just call it the enzyme. We will mention coenzyme and cofactors, especially coenzymes. We'll talk about a few coenzymes in this lesson. When we're talking about aerobic respiration, we'll talk about NAD+, this nicotinamide adenine dinucleotide, a niacin derivative, which is involved in catabolic reactions primarily. And what we'll talk about is it's involved in aerobic respiration. And then FAD, that's flavine adenine dinucleotide, and it too is an enzyme or coenzyme, whatever you want to call it, which is important in aerobic respiration. And that's why we will talk about these two enzymes. The other coenzymes, we're not going to talk much about them, if at all. We won't talk about NADP+. That coenzyme is found in photosynthetic cells, like plant cells. FMN, I think that's actually involved in aerobic respiration, but we're not going to talk about it. Coenzyme A, we will briefly talk about it. It is involved in the preparatory step of the aerobic respiration. But you don't need to know this as a coenzyme. You should know NAD+ and FAD are two important coenzymes that we will talk about in this lesson. Any question about any of that? All right. So this slide is showing you the different, what do you call these, models for how an enzyme works. The first model is the lock and key model, where a specific substrate binds to a specific enzyme, such as a specific key fits one specific lock. And then what happens is the enzyme substrate form a complex, which, let me blow this up, which puts stress on the binds of that substrate, making the substrate change and becoming the products, meaning it'll change the chemical bonds in the substrate. And then the product is released from the enzyme, and then the enzyme is unchanged and then combined to another substrate. And this reaction can happen thousands or millions of times. It's the lock and key model for how an enzyme works. This here is showing you the hand and glove model, of how an enzyme works. And the enzyme is the glove, which fits over the hand, which is the substrate. And what do you notice about this diagram? There's actually three things that can be pictured here. What do you notice about this picture? Come on, I'm going to require you guys to mention the two things. The third, you got to look at really carefully, and most people won't see it. What do you notice about the substrate and the enzyme and the substrate enzyme complex? There's no discussion. We're just going to move on.
The shape of the enzyme changes.
Yes, sort of like the glove molds to your hand to fit the hand. The enzyme does change. What do you notice about the size of the substrate and the size of the enzyme?
Substrate is much smaller than the enzyme.
Yes, the substrate is much smaller than the enzyme. These enzymes are biological molecules and they are very complex. They're very large. And the enzyme molds to the substrate and changes its shape when it's in the enzyme substrate complex. Let me blow this up now so you guys can see the third one. I'm not going to require you to state the third one, but you'll notice how the substrate here and here, they change. The substrate is flatter here than it is there. And this would be the shapeshifter's hand molds to fit the glove when the shapeshifter is putting his hand into the glove. And this would be, I guess, the shapeshifter's hand and glove model. So if you know about Star Trek, you know what Star Trek Deep Space Nine, you know what I'm talking about, where the shapeshifter's hand molds the shape of the hand. Come on, mouse. Shapeshifter's hand molds to fit the shape of the glove, while the glove is molding to fit the shape of the shapeshifter's hand. So the two actually mold to fit each other. I'll word it that way. Any question about any of that? And those of you who are familiar with Star Trek Deep Space Nine, you know what I'm talking about when I'm saying a shapeshifter. All right, I've already stated that enzymes do not work in a vacuum. There are various factors that influence an enzyme activity. The temperature, the pH, and the salinity have to be correct in order for the enzyme to work, at least work at its optimal level. And when the temperature, the pH, and the salinity are not at the optimal levels, the enzyme will not work at its optimal rate or optimal level, if it'll work at all. Heavy metals can also influence the enzyme's activity. Alcohol, UV radiation. All of these, heavy metals, alcohol, and UV radiation, can cause protein denaturation. And if you get the protein totally denatured, the enzyme cannot function. That's actually true for the temperature, the pH, and the salinity. If those are not at the optimal condition, meaning the optimal temperature, or the optimal pH, or the optimal salinity, the enzyme will begin to denature. And way off from the optimal, like boiling temperature, that will totally denature the protein of the enzyme, and then the enzyme will not function. The substrate concentration can also influence the enzyme's activity. Note the substrate concentration does not denature the enzyme, but we'll show you a graph. The substrate concentration can alter the enzyme's activity. Generally, as you increase the substrate concentration, you will increase the enzyme's activity, and that's largely because you increase the substrate concentration, you increase the chance that the enzyme will bind to a substrate and then increase the enzyme's activity. Various poisons can also influence the enzyme's activity. Poisons usually inhibit an enzyme. Cyanide and fluoride are two examples of poisons which kill enzymes. Any question about any of this? All right. We have inhibitors which can influence the enzyme's activity. When you have an inhibitor around, the enzyme will decrease its functioning, so it'll have less activity. And when you have a lot of the inhibitor around, the enzyme won't work at all. There are two different types of inhibitors: competitive inhibitors and non-competitive or allosteric inhibitors. We'll also talk a little bit about feedback inhibition. Here we're looking at an enzyme in its normal three-dimensional folding pattern. That enzyme can be denatured, and this is totally denatured, where the enzyme will not have any activity. For example, if this is the active site right here, binding to the substrate, there is no functioning active site here. So this enzyme cannot perform. And that's totally denatured, where the protein of the enzyme is in a totally different shape. And it will not function when it's totally denatured. Any question about that? Oftentimes, a denatured enzyme is one that has chemical bonds broken. For example, if there's a hydrogen bond between here and here, that's broken here. If there's an ionic bond between this one and that one, it's missing there. So the bonds can be broken, but you could take the enzyme and put it in the refrigerator. And if it's an enzyme that requires 37 degrees Celsius, that's 98.6 degrees Fahrenheit, that will form new bonds that shouldn't be in the enzyme. And that will change the folding pattern of the enzyme, and then the enzyme will not function. Most enzymes that work at 37 degrees Celsius will not work in the refrigerator. Any question about any of that? So the temperature and the pH can cause denaturization, meaning the enzyme to be denatured. And one example of that is, which isn't an enzyme, it's a protein though. When you crack an egg and you take out the egg white, it's this liquid egg white, and it's mostly transparent, translucent at the very least, because you can see through it. But then when you increase the temperature and you cook that egg white, you change the appearance from translucent to opaque white, and then you'll change the egg whites from a liquid to a solid. And what you're doing when you're cooking the egg is you're denaturing the egg white. And most of us eat denatured egg white. There are a few people who do eat functional or active egg white, meaning a raw egg white, but most people don't. I knew somebody who, when he was, joining the Navy, he didn't weigh enough, and so he was adding raw eggs, I assume the egg whites, to the milkshakes to get his weight up, and then he went and joined the Navy, and he got on the scale, and he just barely weighed enough. That's the only person I know who ate egg whites raw, but there are some people who do. There are different factors that can influence the enzyme's activity, such as temperature and pH. This enzyme has an optimal activity level at 37 degrees. where the enzyme will work at its optimal rate. So this enzyme is probably an enzyme like a human enzyme inside the body, working optimally at 37 degrees. And when you move away from 37 degrees, the enzyme begins to be denatured and it loses its activity level. The further you go away from the optimal level, the less enzyme activity you have. And then when you go way away from the optimal temperature, which in this case would be 50 degrees Celsius or 10 degrees Celsius, the enzyme loses all activity. At these temperatures, the enzyme is totally denatured. And that would be a enzyme in the refrigerator. I'm not sure what 50 degrees Celsius, what temperature that is. It's much less than boiling, but this enzyme stops working above 50 degrees Celsius. And it's totally denatured at that temperature. Any questions about temperature influencing the enzyme? All right, the same thing happens with the pH. Around a pH of 5, this enzyme has an optimal activity level, meaning the enzyme works best around pH 5. And then when you move away from pH 5, the enzyme's activity level drops. The enzyme is slightly becoming denatured here, meaning the change is to the enzyme is it's unfolding, but it's not totally unfolded. And then when you move to extreme pH or a pH 0, in this case, or a pH 10, the enzyme is totally denatured, totally out of its three-dimensional shape, and the enzyme will not function. Where do humans have a pH of 5, where this enzyme would have its optimal activity? Where do humans have this? Anyone? Where does humans have a pH of 5?
Our stomach?
Can you say that again? I didn't hear you.
Our stomach?
No, our stomachs go down to about pH 1 or pH 1.5. So that would be much too low for this enzyme. But that's a good guess.
Or blood?
No, our blood is around pH 7.4, and our blood would have to be close to pH 7 for us to stay healthy. Our blood is not acidic. There might be an exception in the lungs because we're breathing in oxygen and breathing out CO2, and there might be a little bit more.
Our urine?
You know, our urine is around pH 5. That is true. I'm not sure exactly what pH the urine is, but it is slightly acidic, and that would be a good example. I'm not sure what the pH of the urine is, but it's close to that. That's not what I was trying to get at. I imagine we do have some enzymes, like in the bladder, which do work in a slightly acidic environment. I'm not aware of any enzymes that we put into the urine, because the urine is something that we're going to void from the body. And so I don't believe the body puts an enzyme to change something in the urine, but I could be wrong. I simply might not know about it. But no, I was trying to talk about the skin. The skin is slightly acidic around pH 5. We'll talk a little bit about that later. Come on, mouse. And part of the reason why the skin is acidic is we make it slightly acidic so that if a pathogen gets on our skin, it might not be able to grow and get established because it's too acidic. And then sebum itself is slightly acidic, but our own microbiota, the bacteria and microbes of our skin, digest that sebum and make it even more acidic, lowering the pH to around pH 5. So this enzyme is likely on the skin, although it could be the bladder. And I'm gonna take a look there and find out what the pH of the urine is.
I think it's usually between like 5 and 8 at work when we dip someone's urine.
Uh-huh. It would be very acidic, but if you're eating a lot of vegetables, it might be. Yeah, it's between 4.5 and 8. Typically, it's around pH 6, but it can range from 4.5 to 8. And it would be alkaline if you're eating a lot of vegetables, which are alkaline, and the body is partly trying to normalize the pH of the blood. And so it's putting out the bases in the urine. All right, let me share my screen again. All right, the substrate concentration can affect the amount of enzyme activity. When the substrate is very low, the enzyme's activity will be very low because the chance of an enzyme finding a substrate and then converting into the product is just low because there's not much substrate around, so the two aren't going to come together very often. And so the enzyme will have a low activation rate. But as we increase the substrate concentration, we dramatically increase the activity of the enzyme up until a point, meaning the substrate concentration, as we add it, the activity level starts to lessen. And then eventually we get to the point where when we increase the substrate concentration, it does not increase the activity of the enzyme at all. And the reason for that is at this point here, all of the enzyme is already bound to a substrate. And so if we increase the concentration of the substrate, you can't increase the activity of the enzyme any faster because at this point, all the enzyme is already bound. And that's why when you increase the substrate concentration, it takes a graph like this. Initially, it increases the enzyme activity very rapidly, and then it starts to slow, and then there's no increase in the enzyme's activity. Any question about any of that? All right, so the big point here is enzymes do not work in a vacuum. They do require an optimal environment, like an optimal temperature and optimal pH to work, at least at their optimal level. And then some enzymes also have a helper. So if the enzyme requires the helper, that helper must be there for the enzyme to function. We can have an inhibitor around, which also influence the activity of an enzyme. An inhibitor inhibits the enzyme. There's two types of inhibitors. There's competitive inhibitors, and they compete for the active site of the enzyme with the substrate. And if we have a drug, which is a competitive inhibitor, if you want to shut down the enzyme, you just make sure the concentration of the drug is higher than the concentration of the substrate, and then the drug will bind to the enzyme. And once it's bound to the enzyme, that enzyme cannot bind to the substrate. And that's why it's called competitive inhibition, and that is the inhibitor is competing for the active site with the substrate. Any question about any of that? All right, the other type of inhibition is non-competitive inhibition, which is also called allosteric inhibition. Here we have an enzyme which binds to its substrate, and we have an inhibitor which also binds to the enzyme. The non-competitive inhibitor does not bind to the active site of the enzyme. It binds to another site of the enzyme. And this site is called the allosteric site. Why this is called the allosteric inhibitor, as well as a non-competitive inhibitor. When the allosteric inhibitor binds to the enzyme, it changes the folding pattern of the enzyme. So the enzyme will now take on a different folding pattern, and that will change the active site. And this active site can no longer bind to the substrate. And so that will inactivate this inhibitor enzyme. Any question about non-competitive inhibition? If not, let's talk about feedback inhibition. This is where we have an enzymatic reaction converting a substrate into an intermediate molecule by enzyme 1. And then we have enzyme 2 taking the intermediate A to make intermediate B. And then we have enzyme 3 to take intermediate B to make the end product of the biochemical reaction or the pathway of the reaction. Feedback inhibitor usually involves the end product that's made binding to the first enzyme and then shutting down that enzyme by non-competitive inhibition. This is frequently seen in cells because it is a very efficient way to control how much end product the cell will make. And that is when there's very low amounts of end product and the cell wants more, the end product isn't around to bind to the enzyme, and so enzyme 1 will be active, making intermediate A, enzyme 2 makes intermediate B, enzyme 3 then makes the end product. As the end product starts accumulating in the cell, this end product will begin binding to enzyme 1, shutting it down. But there will be other enzyme 1 which will be active, so the cell will continue making end product until the concentration of the end product reaches a high level. And when the end product is at a high level, it will bind to enzyme 1, shutting enzyme 1 down, and then all of enzyme 1 will be shut down. No enzyme 1, we make no intermediate A. No intermediate A, we make no intermediate B. No intermediate B, we make no more end product. So the cell will not get make any more end products. The concentration in the cell will no longer increase. And then with time, let's assume the end product is being used up. There will be less and less end product. And that means that when an end product falls away from the enzyme 1, there won't be another end product around to bind to enzyme 1. And so enzyme 1 will now start making intermediate A, making intermediate B, and then making more end product for the cell. And that's how a cell uses feedback inhibition to keep the level of the end product within a certain range. Whenever the end product concentration gets lower than that range, there won't be enough end product around to shut down the enzyme. And when enzyme 1 is active, we'll start making more end product. And then when there's more end product made in the cell, and there's enough of it, it will bind to all of enzyme 1, shutting the enzyme down. And then there's no more end product made in the cell. Any questions about feedback inhibition? All right, I've already mentioned that feedback inhibition like this is frequently seen in cells, and that's because it's a very energy efficient way of keeping the end product within a certain range. And it's also energy efficient because you shut down enzyme 1, not enzyme 2 or enzyme 3. If you were to shut down enzyme 2, then intermediate A would be made in the cell, and that will take the cell energy to make intermediate A. But then intermediate A is not going to be used to make the end product. And so we're making intermediate A for no purpose. And that would be wasteful of energy for the cell. So it's best to shut down enzyme 1 by the end product itself. And then you control how much end product is made, as well as shut down the first enzyme. And that's the most energy efficient way to control this biochemical reaction. Any questions about feedback inhibition? All right, I've implied or even stated that enzymes tend to be proteins. Most enzymes in cells or anywhere are protein, but not all enzymes are made of protein. Most are, but not all. There are some enzymes which are made of RNA, and these have a specific name, a ribozyme. It is an RNA molecule that cuts and splices RNA or some other molecule, much like an enzyme would cut and splice RNA or some other molecule. These RNA enzymes function like enzymes in that they have an active site that actually binds to the substrate, and then the ribozymes are not used up or modified in a chemical reaction. So once this ribozyme performs its chemical reaction, it will release the product, and then that ribozyme can bind to another substrate and perform another biochemical reaction. All right, this is the only time I believe Tutora talks about ribozymes in the textbook. He does not mention it, but we're going to talk about aerobic respiration. No, sorry, not aerobic respiration. We're going to talk about protein translation. And in protein translation, we functionally take the growing polypeptide molecule, meaning the growing polypeptide that will become the functional protein. When we're adding a new amino acid, there's an enzyme that removes the growing polypeptide and then adds it to the latest added amino acid in protein translation. This enzyme is actually a ribozyme. It's the ribosome that does this, and it's the RNA portion of the ribosome that does that. And so that enzyme is the one case of an RNA enzyme that we're going to talk about in the class. And Tutora doesn't mention it, but I just did. That enzymatic reaction happens by an RNA enzyme. And it's simply the removal of the growing polypeptide from its attached tRNA, and then you bind that growing polypeptide to the new amino acid that was just added. And at the moment, that amino acid is still attached to the tRNA. You don't need to know about the tRNA part. You just need to know that that's the one RNA enzyme or ribozyme that we will actually talk about. All right, any question about any of that? So most enzymes are proteins, the vast majority, but there are a few which are made of RNA. If there's no questions, I'm going to move on to metabolic diversity among organisms. This is a new topic, but we can say all organisms, including microbes, require the following. They require an energy source and they require a carbon source. We can classify an organism based on where they get their energy source from. Organisms are classified as a phototroph if the organism derives its energy needs from light. Phototroph meaning light, troph, whatever troph stands for. I don't remember. I used to know. Let me see if I can recall. I think troph has to do with a living organism. So light, get their energy needs from light, phototroph. Chemotroph are organisms that derive their energy needs from an inorganic or organic chemical compound. We are a chemotroph, and we get our energy needs from an organic chemical compound, mainly glucose, but it can be any food molecule that we digest. So we are a chemotroph. There are some chemotrophs which can derive their energy needs from getting their energy from a chemical compound, an inorganic chemical compound, such as sulfate or nitrate or nitrite or iron, or what's another example? Hydrogen sulfide gas. And we'll talk a little bit about those. Those are actually the chemosynthetic bacteria. They are chemotrophs, deriving their energy needs from an inorganic chemical compound. We can also classify organisms on where they obtain their carbon source from. Organisms are classified as an autotroph if they get their carbon from CO2, either in the air or the water. Green plants are an autotroph because they pull CO2 directly from the air. We are a heterotroph because we get our carbon needs from an organic chemical compound that we consume, such as glucose. Any question about any of that? All right. You can then talk about the diversity of organisms and consider both the energy and the carbon source so that an organism can be defined as one of these four groups: a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. This table is showing you the diversity among organisms, separating them into the four classifications. The photoautotrophs are an easy one to talk about because everyone knows green plants, and we know they get their energy needs from light. That's why they're a phototroph. And they get their carbon source from CO2 in the air. That's why they are an autotroph, and they are a photoautotroph. You should know that there are bacteria, which are photoautotrophs, such as the cyanobacteria. Obviously, green plants are photoautotrophs. And both of these organisms are oxygenic photoautotrophs, meaning they make oxygen in photosynthesis. But photosynthesis does not need to make oxygen. And we have anoxygenic photoautotrophs. They make sulfur, and the green and purple sulfur bacteria are an example. You don't really need to know about the green and purple sulfur bacteria. I'll never quiz you on that. The only point is you should know that the photoautotrophs can make oxygen, but they don't have to. Green plants, of course, do make oxygen. The next simplest to talk about are the chemoheterotrophs, And this is simple for students because we are a chemoheterotroph. Chemoheterotrophs get their energy source from a chemical compound, such as glucose, and they get their carbon source from an organic chemical compound, such as we get our carbon typically from glucose. There's more than just humans, which are chemoheterotrophs. The fermentative bacteria, like the bacteria used to make yogurt, are chemoheterotrophs. All animals, protozoa, all fungi, and then many bacteria are chemoheterotrophs. Like E. coli is a chemoheterotroph. They get their energy needs from glucose or some molecule similar to glucose, and they get their carbon source from an organic chemical compound, largely from glucose. And so the E. coli are bacteria like us. The fungi are like that as well. There are some bacteria which do get their energy needs from a chemical, but that chemical is not glucose. It is an inorganic chemical. And what they are doing is they're stripping the electrons from this inorganic chemical compound and then using those electrons to serve the cell's energy needs. It's the chemosynthetic bacteria which can do that. They get their energy needs from a inorganic chemical compound, such as iron or nitrite, or any of these other inorganic compounds. The next easiest to talk about are the chemoautotrophs. They get their energy needs from a chemical. This is almost always an inorganic chemical compound, such as iron or nitrite or hydrogen sulfide gas. These organisms are the chemosynthetic bacteria, obtaining their energy needs from an inorganic chemical compound, and these organisms get their carbon from CO2. Why they are a chemoautotroph? Because they're pulling their carbon from CO2. You don't probably know this, but you are familiar with a chemoautotroph. And that is if you lift up the back of the toilet, meaning the toilet tank, not the toilet bowl, but the tank, you can see this brownish or yellowish scum on the side of the bowl. That is a chemoautotroph bacteria obtaining their energy needs from stripping electrons from iron. The iron is dissolved in the water, and then the bacteria are getting their CO2 needs from carbon dioxide dissolved in the air or the water. So there, you actually are familiar with the chemoautotroph, the brownish or yellowish scum on the toilet tank. Now, let me warn you that the chemosynthetic bacteria, some of them are chemoautotrophs, but some of them are chemoheterotrophs. The difference is if this chemosynthetic bacteria gets its carbon from CO2, that's a chemoautotrophic chemosynthetic bacteria. However, if the bacteria gets its carbon needs from an organic compound such as glucose, then this is a chemoheterotroph chemosynthetic bacteria. Any question about any of that? I don't know an example of a chemosynthetic chemoheterotroph other than maybe euglena, which isn't a bacteria, but they do exist. Any question about any of that? And I just gave you one complication of this thing, and that is the chemosynthetic bacteria may be a chemoautotroph, but it may be a chemoheterotroph, and it will depend on where they get their carbon. All right, the hardest group for students to understand are the photoheterotrophs. They get their energy needs from light, why they are a phototroph, and they get their carbon from an organic chemical compound such as glucose. And that's why they are a heterotroph. You're not familiar with any organisms that are photoheterotrophs, but I will tell you the green and purple non-sulfur bacteria are photoheterotrophs, obtaining their energy needs from light and their carbon from an organic chemical compound such as glucose. Any question about the metabolic diversity among organisms? If you get confused on this, just go back to say, are they getting their energy needs from light or from a chemical? They're a phototroph if they derive their energy from light, a chemotroph if they derive their energy from a chemical compound. And then in the other back is back to where they're getting their carbon source from. They're an autotroph if they get their carbon from CO2, a heterotroph if they use an organic chemical compound such as glucose. And then once you have that, you can develop the grouping by both the carbon source and the energy source. This is another looking at the classification of organisms. You should know the organisms in red. Let me blow this up. So first we have all organisms. We're mainly talking about microbes in this class. So we're talking about all microbes. And then we can split them on where they get their energy source. If they get their energy needs from a chemical compound, then they're a chemotroph. If they get their energy needs from light, then they're a phototroph. We can then split the phototrophs depending on where they get their carbon source from. If they get their carbon from an organic chemical compound such as glucose, they are a photoheterotroph. If they get their carbon from CO2 in the air or the water, then they are a photoautotroph. We could go further and say that photoautotrophs can make oxygen or they do not make oxygen. I'm not going to require you to know this part for the quizzes and exams, so you don't need to know it. We can also split the chemotrophs on where the organism gets its carbon source from. If they get their carbon from CO2 in the air or the water, then they are a chemoautotroph. One chemoautotroph that you are familiar with is that yellow or brown sludge in the back of your tank. And most of that is a iron oxidizing chemosynthetic bacteria. If they chemotrophs get their carbon source from an organic chemical compound, such as glucose, then they are a chemoheterotroph. You may split the chemoheterotrophs onto where the final electron goes, meaning what is the final electron acceptor. If the final electron acceptor is oxygen, and that's all animals, including humans, most fungi, protozoa, many bacteria, and these are chemoheterotrophs, which are performing aerobic respiration, where the electrons end up at oxygen. We'll talk a little bit more about that later, but you don't need to know that for this slide or for the quizzes. You can also split the chemoheterotrophs into the organisms that do not perform aerobic respiration, and these we call non-aerobic respiration. All right, any question about any of that? Let me go back one slide and say that I need to talk about one other thing, and that is with the chemoheterotrophs, many organisms get both their carbon and their energy source from the same molecule. For example, humans mostly get their carbon and their energy needs from glucose. And if it's not from glucose, it's from some other molecule of our food that we consume. But not all chemoheterotrophs require that. All right, any question about the trophs? If not, let's talk about energy being produced. Cells need to make energy and then store that energy in ATP. We'll talk about ATP in a little bit. But where the energy comes from is the nutrient molecules we eat contain energy associated with the electrons in the bonds within that molecule. So for example, when we eat glucose and then we digest the glucose to get our energy needs, metabolize the glucose, I guess would be a better word. We break down the chemical bonds in glucose and then harvest that energy and store it. We actually store that energy in ATP. And this is often seen in cells where the energy is obtained from a catabolic reaction, where the energy from a complex molecule is obtained by breaking down the molecule, such as glucose in aerobic respiration. The energy in the carbon links of glucose is used by the cell to serve its energy needs. Most of that energy will be converted into the energy of ATP, meaning we make ATP. And then when the cell needs energy, it will break down ATP to release the energy and then use that energy for the cell's energy needs. ATP is called the energy carrier of cells because it has a high energy bond, which is unstable. And once the cell breaks that bond, that releases a lot of energy, which the cell can then use for the cell's work. Any question about energy production or ATP? We will talk more about ATP in this lesson, but for now, let's move on. Energy being produced by cells, if they do catabolize a molecule to get their energy needs, then they do that by oxidation reduction reactions. Oxidation is the loss of electrons. So molecule A here is going to lose its electrons, or in this case, one electron. So molecule A will be oxidized because it's losing its electron. Molecule B, on the other hand, is going to gain the electron. Gaining an electron is reduction. And so molecule B will gain the electron, so molecule B will be reduced. Now you can remember this, that oxidation is loss of electrons, reduction is gain of electrons, is with this mnemonic, whatever. And you can say, Leo the lion goes grr. Leo, lose electrons, oxidation. Leo the lion goes grr, grr. Gain electrons, reduction. So a molecule that loses its electron will be oxidized. The molecule that gains the electron will be reduced. And this is an actual example. We have copper +1 plus iron with 3 positive charges can be converted into copper +2. Copper is being oxidized, losing an electron. And then iron will be +3, will be gaining the electron, and it will be reduced. It'll go from iron +3 to iron +2. Any question about any of that? In biological systems, oxidation usually has the hydrogen ion following the electrons. Remember, in a hydrogen atom is made of one H plus ion and then one electron. And if you have the hydrogen ion lose its electron, then the hydrogen atom will be oxidized, and whoever gains the electron will be reduced. Anyways, because the hydrogen ions are frequently following the electrons, we call these dehydrogenations, where this molecule loses an electron, but it also loses a hydrogen ion, and that molecule here is gaining the electron, and it's also gaining a hydrogen ion from this molecule. And we'll actually talk about NAD plus gaining an electron and a hydrogen ion to become NADH when we talk about aerobic respiration. All right, biological redox reactions are often used in catabolism to extract energy from the nutrient molecules. Humans and almost all chemoheterotrophs perform catabolism to get their energy needs. And how they do that is they begin with a highly reduced compound. That's a compound with many hydrogen ions. And then they degrade that compound, catabolize it to make it a highly oxidized compound. For example, when glucose C6H12O6, and you should know that if you don't already know it, is a highly reduced compound with many hydrogen ions, and then that becomes oxidized to carbon dioxide and water. Along the way, that glucose will be used, the energy will be taken from the glucose and used to make ATP. So we can say that these molecules that are being reduced for obtaining our energy needs have a large amount of potential energy in them, potential energy being the energy in the bonds of the chemical reaction of glucose. And if we break those bonds in glucose, we can then release that potential energy and use that energy to make ATP. Any question about any of that? All right, here we're looking at aerobic respiration, which is the energy conversion in a cell, where glucose plus oxygen can be converted by the cell into carbon dioxide and water. Along the way, ATP is generated in aerobic respiration. You'll note that this slide is trying to show you that aerobic respiration is the same thing as cellular respiration. The two are used interchangeably, meaning they are the same term. I don't tend to use the word cellular respiration because a bacteria that is engaging in chemoheterotroph, engaging in breaking down a molecule to obtain its energy needs and its carbon needs, such as breaking down glucose, is a cell, and it may perform respiration, but that respiration doesn't have to be aerobic respiration. For example, E. coli can break down glucose into CO2 and water using aerobic respiration. However, if oxygen is not around, E. coli can switch using aerobic respiration to using anaerobic respiration. And that too will degrade glucose and generate energy, but this will create less energy than aerobic respiration. And I don't want to focus on that. All I'm trying to focus on right now is that cellular respiration literally means the same as aerobic respiration. I don't like the term cellular respiration because you can have a cell engaging in respiration, but it is not aerobic respiration, such as when E. coli is performing anaerobic respiration. Any question about any of that? All right, so the summary of aerobic respiration is shown here. We start with glucose, C6H12O6, add on 6 oxygen molecules, and you have 38 ADP and 38 inorganic phosphate. That's what the PI stands for. And then you make CO2, water, and then you make ATP from ADP. And that's the summary reaction of aerobic respiration. Meaning this is one step where we're discussing aerobic respiration. In reality, aerobic respiration engages in many chemical reactions and then some physical reactions. So aerobic respiration does include many chemical reactions and then some physical reactions. Here we're looking at energy production, how cells can generate ATP. Some cells can generate ATP by phosphorylation, meaning you take ADP and phosphate, PO4 with the -3 charge. You need to add on energy to get this reaction to occur, and then you make ATP out of it. So ADP plus energy and phosphate make ATP. When we look at ATP, there are 2 bonds in it, which represent a high energy bond. And when the cell needs energy, it can break this 3rd phosphate off, and that will release the energy in that bond. And the cell can use that energy for its energy needs. If the cell needs further energy, it can break the 2nd phosphate off, and that too is a high energy bond. Any question about making ATP? Cells have 3 mechanisms for making ATP. The first is substrate level phosphorylation. This is where you transfer a phosphate group from some phosphorylated molecule, and that's going to be abbreviated CCC with a phosphate group attached to it. You can break that phosphate off and add it directly to ADP, and then you get ATP and then the 3-carbon molecule without the phosphate. This actually happens in fermentation and glycolysis, and so we'll look at that in, well, probably not today, but later in this lesson, which will probably be Wednesday. The second way cells can generate ATP is by respiration. This involves a sequential transfer of electrons from the electron donor cell, which is usually glucose, to the final electron acceptor, which for aerobic respiration, the final electron acceptor is oxygen. Along the way, that electron is moved from one molecule to another, to another, to another, and initially the electron flows from glucose, moves to NADH, moves from NADH to FMN, and then these molecules will be reduced, lose the electron, and then the next molecule will be oxidized. Do I have that right? No, lose the electron is oxidation. So if you lose the electron, this molecule will be oxidized, and then Q will gain the reduction. It will be reduced, and then the electron will move to the next molecule, 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 and then finally the electron will flow to the final electron acceptor, the last molecule to gain the electron, which for aerobic respiration, that molecule is oxygen. Oxygen will take the electrons and the hydrogen ions, and then the oxygen will be converted to water. And that's how cells make ATP in respiration. Here we're showing you aerobic respiration, but I could show you anaerobic respiration, and it's similar. We have still all the steps where the electron is transferred to. The main difference is the electron will not flow to oxygen as the final electron acceptor. In anaerobic respiration, the electron will flow to some other molecule that is not oxygen. And it really depends on what form of anaerobic respiration we're talking about, which molecule will be the final electron acceptor. Any question about any of that? All right, the third way that cells can generate ATP is by photophosphorylation. Photophosphorylation is only found in photosynthetic cells. Light releases the electrons from chlorophyll. That light energy is then converted to chemical energy by an electron transport chain, and we'll talk about that at the end of this lesson. ATP and NADPH are generated by photophosphorylation. However, the ATP and the NADPH are often used to make sugar. And so the energy in light will be converted into the energy in sugar. And here we're seeing some photosynthetic organisms. That's why the boys are so muddy. Only kidding. You can see the photosynthetic organisms in the background, though. All right, the energy released from molecules is controlled and stored in a series of reactions. The energy is not released in a single burst. And here we're seeing some of the reactions going on where the electrons from glucose flow to NADH, and then they flow to one other molecule after another. At each transfer of the electrons, the energy in that electron, some of that energy is released. And that's why the cells can perform aerobic respiration at body temperature, 37 degrees, because the energy being released from glucose is not released all at once. The energy is released in small steps. You could release all of the energy at once. Like if I had this cup of glucose here, I could release all the energy by taking a blowtorch to it and converting the glucose into CO2 and water. The energy would be released in a single burst, and I'd have to step way back because of all the energy being released. It'd be too hot and too bright for me to stay close to the cup. How the energy is released is the electrons from donor molecules are passed from one compound to another in a series of redox reactions, as shown here. Most organisms use as their primary source for cellular energy some carbohydrate, and then they use catabolism of that carbohydrate. The most common energy source is glucose, but we and other organisms can use lipids and proteins, and then can catabolize the lipid or the protein to generate energy. But glucose is the preferred molecule. To produce energy from glucose, microorganisms use two general processes to make ATP. There's respiration, and that can be aerobic respiration, or there's anaerobic respiration. The difference is aerobic respiration uses oxygen, anaerobic respiration uses a molecule other than oxygen. The other general way that cells can catabolize glucose or some other carbohydrate is to engage in fermentation. And both ways can utilize glucose to make ATP. Okay? Any questions here? If not, I'm going to end it here. And I'll see you in the lab. Questions? If not, I'll see you in the lab.